Hey guys, I'm back with a new video. Hit the subscribe button and join the club. The topic for today is cell junction and its type. Physiology playlist is available. The link is in the description. So first, what is cell junction? Consider these two cells. The cells are surrounded with a fluid called ECF, that is known as extracellular fluid. So the cell junction is the connection between neighboring cells, or it is the contact between the cell and the extracellular matrix. The cell junction is also known as membrane junction or intracellular bridges, as you can see in the image. Cell junctions are mostly present in immobile cells. They are absent in sperm and macrophages, as they are mobile cells or movable cells. Cell junction is classified into three types. The first is occluding junction, second communicating junction, and third anchoring junction. In this video, we'll discuss these three types of cell junction. So let's start with the occluding junction. Occluding junction is the cell junction which prevents intracellular exchange of substances. That is, this junction prevents the movement of ions and molecules from one cell to another cell. Tight junction falls in this category. The image here gives the brief idea of what could be a tight junction. So the blue colored structure are the cells, and you can see the black structure in the apical region is the tight junction, which is enlarged on the right hand side. The tight junction is made up of protein complexes, nearly 40 different types of protein, which we are going to discuss in a while. So, tight junction. As you all know, it is an intracellular occluding junction that prevents passage of larger substances. It's also known as zonular occludence because it is present in the region where the cell membrane of adjacent cell fuses together firmly. Now, where you can see such tight junction. So this type of junctions are present in the apical margin of epithelial and endothelial cells in the intestinal mucosa, wall of renal tubules, capillary wall, and choroid plexus. Now discussing the structure of tight junction. Tight junction is made up of ridge which has two halves. One half of the ridge is from the one cell and the other half is from the other cell. Both halves of the ridge fuses with each other to form the tight junction. Each half of the ridge consists of tight junction strands that are made up of proteins. So the protein of tight junction are classified in two types. First is the tight junction membrane protein or integral membrane protein or transmembrane protein, such as occluding, clotting, and junctional adhesion molecule JAMs. And the second type is the scaffold protein or peripheral membrane protein or cytoplasmic protein, such as Singulin, Simplekin, and ZO1, 2, and 3. Tight junction membrane protein molecules are anchored in strand of ridge and attached with the countering parts of the neighboring cell so that the cells are held together. The scaffold protein are attached with the tight junction membrane protein and strengthen the anchoring of the ridges. I know that this topic is a bit confusing, so to make you understand, let's use simple diagrams so it can make sense. So here is cell 1, cell 2, orange denotes cell 1, green denotes the cell 2. So this is the ridge. What exactly ridge is? Ridge is a bump or crest, as you can see in the image. So this ridge is made by the anchoring of tight junction membrane protein of one cell with the tight junction membrane protein of the another cell. So here the ridge is made. Now due to this ridge are made up of strands. So the scaffold protein which makes the platform are anchored with the tight junction protein of the respective cells, so they can strengthen the strands of the ridge. To understand this in the better way, let's see the diagram here, which is a realistic one. So you can see the red colored protein denotes the occludins, whereas the orange denotes claudins. These are anchoring and making the ridge, and beside them there is ZO12, ZO123 and cingulins, which are making the platform and anchoring the actin filaments later on. So this is the structure. So basically you can say transmembrane protein or the tight junction membrane protein makes up the ridge, and the scaffold protein or the peripheral protein are anchored with the tight junction membrane protein of the respective cells. Moving on, let's study the function of tight junction. The first is strength and stability. The tight junction holds neighboring cells of tissue firmly, so provides strength and stability to the tissue. Second, selective permeability. The tight junction forms a selective barrier for small molecules and a total barrier for large molecules. Third, fencing function. Tight junction prevents the lateral movement of protein and lipids in the cell membrane, thus act as a fence. This fencing function maintains different composition of protein and lipid between the apical and basolateral plasma membrane domains. Due to this function, the tight junction is sometimes referred as impermeable junction. The fourth is maintenance of cell polarity. Fencing function of tight junction maintains the cell polarity by keeping the protein in the apical region of the cell membrane. Fifth, helps in the formation of blood-brain barrier. Tight junction in the brain capillaries forms the brain-blood barrier, which prevents the entry of many substances from capillary blood into the brain tissues. Moving on, we have defects that are caused by the mutation of genes encoding proteins of tight junction. The first is hereditary deafness. Second is ichthyosis. Third is sclerosing cholangitis, that is inflammation of bile duct. Fourth is hereditary hypomagnesemia, low level of magnesium in the blood. Fifth is synovial sarcoma, that is soft tissue cancer. So it was all for the tight junction. Now moving on to our second category of cell junction, that is communicating junctions. The communicating junctions are the cell junction which permits the intercellular exchange of substances, meaning this junction permits the movement of ions and molecules from one cell to another cell. Gap junction and chemical synapse are the type of communicating junction. So studying the first one, that is the gap junction. Gap junction is the intercellular junction that allow passage of ions and smaller molecules between cells. It is also called as nexus. This type of gap junctions are present in heart, basal part of epithelial cells of intestinal mucosa. Moving on to our next slide, that is the structure of gap junction. Membranes of two adjacent cells are very close to each other. Cytoplasm of the two cells is connected by the channel formed by the membrane of both the cells. So the molecules move from the one cell to another cell directly through this channel without having contact with the extracellular fluid. Each channel consists of two halves. Each half belongs to one of the adjacent cells. Each half of the channel is surrounded by 6 subunits of protein which are called connexins or connexons. Next topic, regulation of diameter of channel of gap junction. Each channel of gap junction is regulated by the intracellular calcium ion. When the concentration of calcium ion increases, the protein subunit of connexins surrounding the channel come close to each other by sliding. Hence, decrease the diameter of channel. The diameter of channel is also regulated by pH, electrical potential, hormones, and neurotransmitters. Moving on, what are the function of gap junction? The diameter of channel in the gap junction is about 1.5 to 3 nanometer. So, this channel permits the passage of glucose, amino acids, ions, and other substances. Second, it also helps in the exchange of chemical messengers between cells. Third, it also helps in the rapid progression of action potential from one cell to the another cell. Studying the defects that are caused due to the mutation in the gene encoding the connexins protein. The first is deafness, the second is keratoderma, the third is cataract, fourth is peripheral neuropathy, meaning damages to the nerves of peripheral nervous system. The fourth is sarcopterygosis, it is a form of neuropathy. And the sixth is heterotaxia, that is abnormal arrangement of organs or parts of body. Second type of communicating junction is chemical synapse. It is the junction between the nerve fiber and the muscle fiber or between two nerve fibers. Through this junction, signals are transmitted by the release of chemical transmitters. Example, neuron synapses. Moving on, let's study the last type of cell junction, that is the anchoring junctions. Anchoring junctions are the junctions which provide strength to the cell by acting like mechanical attachments, meaning this junction provide firm structural attachment between two cells or between a cell and extracellular matrix. You can say anchoring junctions are responsible for the structural integrity of tissues. These junctions are present in heart muscles and epidermis of skin, which are subjected to severe mechanical stress. The attachment between two cells or between a cell and the extracellular matrix is provided by either actin filaments or the intermediate filament, depending upon which type of filament is present in the formation of junction. Junctions are classified into four types. First is the actin filaments junction, which has two subtypes. The first is adherens junction, that is cell to cell. Second is focal adhesion, that is cell to matrix. And the second type is intermediate filament attachment, that has subtypes. First, desmosome, that is cell to cell, and the second is hemidesmosome, that is cell to matrix. Let's study this four type in detail. So first here is adherens junction. Adherens junction is the cell to cell junction which connects the actin filaments of one cell to those of other. The membrane of adjacent cells are held together by transmembrane protein called cadherins. Adherens junction provides strong mechanical attachment to these adjacent cells. It is present in the intercalated disc between the branches of cardiac muscle. During contraction and relaxation of heart, the cardiac muscle fiber are held together by the means of this junction. They are also present in the epidermis of the skin that helps it to withstand mechanical stress. The second type of anchoring junction is the focal adhesion. Focal adhesion is the cell to matrix junction which connects the actin filaments of the cell to the extracellular matrix. In the epithelia of various organs, the junction connects the cell with the basal lamina. The transmembrane protein here is integrins. Moving on towards the third type, that is desmosome. Desmosome is a cell to cell junction where the intermediate filaments connects two adjacent cells. It is also known as macula adherens. The membrane of adjacent cell are thickened and become spot-like patches. Intermediate filaments are attached with these thickened patches. Some of these filaments are parallel to the membrane and others are arranged in a radiating fashion. Desmosome function just like tight junction. The transmembrane protein involved in desmosome are mainly cadherins. The fourth and final type of anchoring junction is hemidesmosome. Hemidesmosome is cell to matrix junction which connects the intermediate filaments of the cell to the extracellular matrix. This type of junction is like half desmosomes, as thickening of membrane of only one cell occurs. As you know, hemidesmosome is between cell and the matrix. So only one cell is present. So only one cell membrane will get thickened like the patches. So it is also known as half desmosomes. Mostly the hemidesmosome connects the cell with their basal lamina. The protein involved here are integrins. So this was all about the anchoring junction. The last point of discussion here is talking about the dysfunctions that are caused due to mutation of genes that codes for the particular cadherins and integrins protein. The first, dysfunction of adherens junction and focal junction in the colon due to mutation of protein result in colon cancer. It can also further lead to tumor metastasis, that is spread of cancer cell from primary tumor to the other part of body. Second, the dysfunctioning of desmosome cause bullous pemphigoid, that is an autoimmune disease and that creates antibodies against cadherins. The third, dysfunctions of hemidesmosomes also cause bullous pemphigoid. In this disease, antibodies against integrins can be seen. That's it for today. This was all about cell junction. Hope you like the video. If you like it, hit the like button and subscribe to my channel. I'm also available on Instagram where I post recent MCQ question regarding medical topic. So follow me there. Thank you for watching and see you in the next video.